Welcome to a new session in molecular biology. In this presentation, we will be looking into the concept of RNA interference. RNA interference it is the process of sequence-specific post-transcriptional gene silencing, extensively found in animals and plants. Usually, the process it is initiated by a double-stranded RNA, which is homologous to the gene to be silenced. Okay, so we can say the RNA interference it is being what you call gene silencing mechanism, right? So the RNA interference it selectively blocks transcription. We can see that the RNA interference it is the mechanism that may inhibit or rarely activate the gene expression at the stage of translation or by hindering the transcription of specific genes. As you can see that the targets for the RNA interference usually include the RNAs from viruses, that is the exogenous pathogenic nucleic acids. Okay, here you can see. So the targets it usually include the RNA which are introduced into the host cell from viruses and also transposons. And these also play an important role in regulating development and genome maintenance. We can see that the RNA interference mechanism is widely used in the treatment of various diseases, including amyloid disease, myeloid leukemia, bone metabolic disorders, and some other such infectious diseases. Now, here you can see the different kinds of RNAs which are used or which are being employed in the RNA interference. The small interfering RNA, yeah, okay. So that is one such kind of RNAs which are very important in the RNA interference process. Similarly, we have the what you call the microRNA or the miRNA, and we have also the shRNA or the short hairpin RNAs because of its shape. So these RNAs, usually the short hairpin RNA and the double-stranded RNA, which later forms the siRNA, these are produced in outside the nucleus, right? It is exogenously produced within the cell, while the pre-microRNA, it comes from, you can see here, the primary microRNA, which is actually the gene transcript of the microRNA gene on the DNA of the cell. Okay, so the microRNA gene, it gets transcribed to form primary microRNA and it is processed to form the pre-microRNAs. The primary microRNA, it gets processed to form the pre-microRNAs or the precursor microRNAs, which gets transported from the nucleus into the cytoplasm. And these what you call the precursor microRNAs, they usually, you can see what is specific about them. Usually the microRNAs, they are single stranded. Here also they are single stranded. It is not a double stranded RNA, but these what you call the precursor microRNAs, they loop back on themselves because there are various regions which do have the complementary sequences. So they loop back on themselves and form a hairpin structure. Even if you can see here, the sequences on this particular stretch, it doesn't have any complementary sequence on the other strand. But still what happens is with a few of the complementary sequences on the strand, they can loop back and appear as double-stranded, right? These precursor microRNAs, it may get acted upon the dicer, which is a protein that can cut these what you call the precursor microRNAs into smaller double-stranded strands over here. So it cuts off the strand, the precursor microRNA over here, and it forms a double-stranded sequence like this. Okay, so this is what actually what you call take part in the RNA interference, and this is what actually complexes with another protein which is known as the risk molecule. Okay, the RNA interference complex protein, and then it forms a risk complex, and this is the one which directly take part in the silencing process. Another one is short hairpin RNAs or shRNAs. Here you can see it is almost similar to the precursor microRNAs having a hairpin loop, and here this is also formed by the looping back of the single-stranded RNA on themselves to form the hairpin structures, and hence looking like a double-stranded structure. The only difference you can see over here, it is slightly longer. In the precursor microRNAs are slightly longer than the short hairpin RNAs. Coming to the next one we have is the double-stranded RNA. It is very unique because the RNAs are usually single-stranded. And these double-stranded RNAs sequences, these can trigger the RNA interference mechanism. And you can see here whether it is a short hairpin RNA or double-stranded RNA or precursor microRNAs, these get broken or processed by the dicer protein, and results in the formation of smaller double-stranded structures which may be like 21 base pairs long, and this is the one which actually take part in the RNA interference mechanism after complexing with the protein complex which is known as the RISC, or commonly referred as RISC complex. Okay, so the RNA molecules which are loaded or which are complexed with the RISC, they can take part in the RNA interference. The first step in the RNA interference is obviously getting bound to the complementary sequences. We'll be looking in detail in the coming slide. Okay, so I hope it is clear how it is happening. Now, so when we discuss about the RNA interference, it is in 1998, Andrew Fire and Craig Mello, they were working on Caenorhabditis elegans. Andrew Fire of Carnegie Institute of Washington and Craig Mello of the University of Massachusetts, they conducted experiments on the nematode. It is a nematode worm, Caenorhabditis elegans. And what they did is they injected these worms with several different preparations of RNA, and they expected to stop the production of a particular muscle protein. Okay, and one of the preparations contained sense RNA. What is sense RNA? That is the RNA which have the sequence of the mRNA that encodes the protein being targeted. Okay, sense RNA means the sequence on the mRNA will be as that of the sense RNA. Okay, just as that of the sense RNA, and it is this sequence which actually codes for the protein. Now, another preparation contained antisense RNA. What is a template? What is a template? The sequence on the template DNA strand. That is a that will be the same as that of the antisense RNA. Okay, so RNA having the complementary sequence of the mRNA. Right, and there was a third preparation which consisted of a double-stranded RNA which contained both the sense strand and antisense RNA. Since they were complementary to each other, they formed a double-stranded RNA. Okay, now what they did is, now what they found is that those nematode worms which were injected with the single-stranded RNAs, that is either sense strand or the antisense strand, in both the situations they did not get much of the effect, right? But in the worms which were injected with the double-stranded RNA, they could stop the production of the particular muscle protein. Okay, so it was very effective in stopping the production of the particular encoded protein. So this particular process of stopping the translation of a particular protein, it was described as the phenomenon of RNA interference by Fire and Mello. And the studies were carried out in 1998, and they got the Nobel Prize for their study on the RNA interference in the C. elegans nematode worm. And they received the Nobel Prize in Physiology or Medicine in the year 2006. Okay, so how these RNA interference actually works? Okay, what is the pathway through which the RNA interference happens? The first step, the RNA interference pathway, it is actually triggered by the enzyme dicer. Okay, we have seen the dicer, which is a protein. So the dicer actually, it cleaves the long double-stranded RNA. Here you can see, even though it's single-stranded, since it has looped back on itself, it has created a double-stranded part of the RNA. So they actually, these dicer proteins acts upon the double-stranded part of the RNA. Okay, so double-stranded RNAs. Now these dicer, they cleave the long double-stranded RNA molecules into shorter fragments of about 20 to 25 base pair length. And one of the two strands of each fragment act as a guide strand, and this guide strand is incorporated into the RNA induced silencing complex, or it is known as the RISC complex. Okay, RNA induced silencing complex. Fine. Now the this can see over here the two strands. It is the double strand, the dicer protein. It creates double stranded RNA molecules of 20 to 25 base pairs length. And what happens later? One of these strands, this gets separated. One of these strands, it gets complexed with the RNA induced silencing complex, or the RISC protein, right? The RNA molecules that have been loaded onto the RISC, they actually form the complex, the direct RNA interfering complex that can actually meddle with the part of the DNA or the messenger RNA, which acts as the target sequence. Okay, so this now we have is the RNA loaded onto the RISC, and this RNA which acts as a which actually complexes with the RISC, it is considered to be the guide strand or the guide RNA. And we can see that they get degraded, the double-stranded the double-stranded RNA, it gets degraded, and followed by this gets complexed with the RISC, and then it forms a complete complex that can bring about the RNA interference. Okay, now this is followed by, the whole process is followed by degradation of the complementary mRNA by the endonuclease Argonaute. So what is Argonaute? Argonaute is a catalytic component of the RISC complex. Okay, so RISC complex, the RNA induced silencing complex, do have catalytic component which is referred as the Argonaute. And we can see that most of the RNA interference, whether it is siRNA or what you call the what you call micro RNAs, we can see that it all do have an Argonaute complex, and this Argonaute complex plays a very important role in bringing about the degradation of the complementary mRNA. Okay, now the short 20 to 25 base pair fragments are small interfering RNA when artificially introduced, or it could be microRNA when it is produced endogenously. What does it refer? That is these double-stranded RNA which brings about the RNA interference after complexing with RISC. So what all are the examples for these double-stranded RNA? It could be the small interfering RNA, or it could be the microRNA. Okay, so this is about the RNA interference, and the phenomenon of double-stranded RNA-mediated RNA interference, it is referred as the RNA silencing. Okay, I repeat, RNA silencing is simply the double-stranded RNA mediated RNA interference. Okay, here what happens is small RNAs, usually complexed with protein machinery, it inhibits the gene expression in different ways. Okay, so here you can see what is the protein machinery. It is the RNA induced silencing complex. Okay, which is the double-stranded RNA that is employed for the RNA interference. It could be siRNA, it could be the microRNA, or it could be the short hairpin RNA. Okay, so it is the double-stranded RNA induced RNA interference is what is referred as the RNA silencing. Clear? Now, here we can see that the RNA interference, it was thought to have evolved as a type of genetic immune system to protect the organisms from the presence of foreign or unwanted genetic material. So if any like exogenous the nuclear material, it could be DNA or RNA, if it enters a cell, a host cell, to get rid of that, this particular RNA interference was used to protect the host cell's own genetic system from the foreign unwanted genetic material. Okay, so this particular mechanism, the RNA interference, it was earlier referred as post-transcriptional gene silencing. Okay, earlier it was also called as post-transcriptional gene silencing or transgene silencing, or it was referred as quelling. Okay, post-transcription gene silencing, the name was common because these particular RNA interference brought about silencing of the gene after the transcription. That is, it worked upon the mRNA. Okay, fine. That is about the RNA interference. Thank you.